like, glad more people can use it. It's also frustrating when suddenly you have to pay for something you were originally covered for.
Yeah.
That's good to know. It's generic at Costco. I love Costco.
Yep, me too. So I'm getting with it, y'all. You know, and especially when I can get cheap medication, and my allergies don't kill me. I have no idea what the tree is that's in my yard. I'm not sure if I'm allergic to it or not. I haven't looked it up yet, but it's dropping all these little yellow flowers all over. My backyard was in a carpet yesterday. My husband and I and my son swept it up. The little yellow flowers, yeah, over my entire backyard. I was like, what? And I'm like, we look up and we're like, they're not all gone yet. It's like, oh, I don't know. But yeah, I'm allergic to ragweed, so next month is really bad for me, which is my birthday month, which is really kind of evil that I'm basically allergic to my birthday. So, yeah. If I had more money, I would maybe go someplace else, but not happening. So speaking of dates and that, so I was looking ahead. We have one more chapter left. It's small. My goal is to get it knocked out next week so that our test will be October 4th. So if you've been procrastinating studying, which I'll pray for you if that's the case, get off your butt now. So I got off my butt, and I did post some Chapter 4 stuff for you guys. I'll probably do another one. I don't know if I'll do it in here or not. I tried to pick the best possible problems, unlike last time. But I did pick one of them, I'll tell you guys. Did I pick it? No, I stole it and put it in my PowerPoint, so we'll talk about it. It was one of the ones where you have to figure out whether you would do product rule or binomial expansion. Remember, I wasn't going to make you guys calculate them. Just tell me what you would do to solve it. So in here, they want you to calculate it, right? So I didn't assign it in Connect. I stole the question and put it in my PowerPoint so we can talk about it. So let's see. So what I want to do today is I want to finish up the material for Chapter 4. And then, with the time left, work through problems. And where we left off last time and add more. So, that is the plan. Having said that, I need to blow my nose. Stop coughing. In here there are tissues. There we go. Much better. So, oh, I didn't pause it. Okay. One other idea I had, because, you know, one of your classmates and I were looking at our poster. We're like, yeah, it's like really small words on that poster. But you know how we were starting to highlight which diseases we talked about. And instead of me doing it, I was like, well, you know, some student could do that as their project. So if somebody wanted to, instead of highlight, what I want to do is I want to start creating a list that we put up on the bulletin board of what disease, what its pattern of inheritance is, like for Huntington's disease, that it's dominant, and then listing what chromosome it's on. So starting, you know, adding to our bulletin board. So if you're interested in doing that, email me. And you know, so some of these we don't know what chromosome, right? So you'll have to research it. And then of course include the link of where you found that. And you can either print it yourself or you can email it to me and I'll print them and start putting them on our board. And more than one person, like 2 or 3 people can work on it, it's fine. But we'll need to, if we have more than one person interested, then we'll have to divvy up the diseases, you know. So whenever I talk about one, I'll be like, okay, whose turn is it to look this one up, right? So email me if you're interested in that, and we'll organize it. So I think that'd be a great project. And then you could probably, you know, go into more detail for one of them for your research project for this class. So just remember, you got to do some type of research project. I'm not talking about some big ugly paper, right? I'm talking about, you know, a PowerPoint presentation or a poster board or, you know, a video recording of you talking. I mean, I'm very open to how you present what you've researched. I don't want to read papers. Most people don't want to write papers, but if that's what you like to do, that's acceptable too, right? But multimedia format is very acceptable as well. So we'll talk more details after that, after test 1 too. But that's something you should be thinking about, you know, what do you want to know more about, right? And then remember, as far as the chapters, that I would like for you to focus in on as it relates to doing research would be the ones I listed on our modules page. So you could pick a disease, but we want it to relate to things like population genetics or medical genetics and cancer. I mean, any disease falls into that category, right? If you want to research a particular disease. If you're interested in biotechnology, you know, you want to do some research into a particular topic in biotechnology. And then you can look at these chapters to get specific ideas as well. And if you're interested in evolutionary genetics, you know, I mean, you could do more about that big, huge Petri dish thing that we talked about. You could, you know, put together something about that research. You know, that's an option. That's evolutionary genetics right there in action. All right. So to our lecture, so part 4, if I could click on the right part, that would be good. So we just have 2 small sections left. This section dealing with pleiotropy. So we need to be able to explain, right, this phenomenon of pleiotropy. So most genes, as we've kind of said, I've kind of said over and over again, right? Most of the things that we talk about aren't controlled by just a single gene, right? Most genes actually have multiple effects, even. So it's not as simplistic as sometimes it appears to be. So multiple effects of even a single gene on a phenotype of an organism is referred to as pleiotropy. So what does this mean? Or how can this be? Well, especially when you're talking about multicellular organisms, right? They're made up of lots of different cell types, right? We're made up of lots of different cell types. So the gene product can affect the cell function in more than one way if you're talking about a particular cell. Or if you're talking about a multicellular organism, that gene could be expressed differently in different cell types. And then the third would be that gene may be expressed at different stages of development even, right? So as the organism is developing, what that gene, right, what product it does and what role that product plays changes as the organism develops. So cystic fibrosis is the example that they use. And this is one that we've talked about previously, right? Did we figure out which chromosome that one was on? I don't think we have yet. So. Yeah. No, because all we have is Huntington's and sickle cell highlighted. So I don't think we figured out where cystic fibrosis is. So that's one of the ones on the list if you're interested, right, in working on it for our genome mapping. So the normal allele codes for cystic fibrosis transmembrane conductase regulator. Oh boy, that's a mouthful, huh? What is that? It's a protein embedded in the membrane that regulates ionic balance. And here we find out, because remember before I said, I don't remember exactly what ion. It's the chlorine ions. And so, unfortunately, the mutant does not transport chlorine effectively. It doesn't not do it at all, but it just doesn't do it as well as it should. Because of that, people who have it, particularly in the lungs, this affects the what's the word? Viscosity of the mucus. It's thick as opposed to being a little bit more watery, right? Because this ion affects the water balance as well, right? Ions affect water movement, osmosis, right? Remember these things, right? On the skin, though, it affects our reabsorbance of salt. So when you sweat, it's salt is also in that water. And there's different theories out there. I haven't done any research into it as to why we actually add salt, but believe that it actually helps in that cooling process because you add salt to ice when you're making ice cream, right? So it's that interaction that helps in the cooling process. And the salt actually kind of melts the ice a little bit even. And you seem like, why would that be beneficial, right? Well, when the ice is melting, it makes more contact with the container to cool the ice cream faster. So for these people, they don't reabsorb some of the salt. So their skin is saltier than most people because they have this problem getting it back in because, right, the transport isn't working efficiently. So that's actually apparently a diagnostic test even that they can do. It's to test how salty your skin is as a preliminary diagnosis for cystic fibrosis. Interesting, huh? If your dog or cat is licking you incessantly, they like salty things. They'll lick you. Mine likes to try and lick the lotion off my legs. I'm like, go away, Joy! You can't have my lotion. I need it. So males are often sterile, unfortunately, if they have cystic fibrosis. And that's because, again, the chlorine ion transportation is really important for the proper development. So this one even affects development of the vas deferens, which is where the spermy guys are stored, right? No, vas deferens. Yeah, that's where they're stored, right? Or is that the epididymis? They mature in the epididymis. But they're stored in the vas deferens. It's been way so long. Yeah, it's the tube. Yeah, it's the tube they travel through, but I think they're stored there too, I think. Getting ready to be called up and go through the tube. That's why apparently you want them to not stay there too long because, then they start not being as good. But anyways. So the defect, right, as we can see, has multiple effects depending on the cell type, right, that is affected and what the job is of that cell and how important that chlorine transport is for those particular cells. So probably the most, detrimental for people and that most people are aware of is that problem with their lungs, right, with having cystic fibrosis. So the next section, we talk about gene interactions. So this is where now genes, again, aren't really standalone things, right? So you have these interactions with one another. So there's different types of interactions. Epistasis. Oh, I always mess this one up. Epistasis. Stasis. Complementation, gene modifiers, and gene redundancy. And as the last one implies, that one's pretty easy to figure out, right? Redundancy means what? Multiple, right? So you have, multiple things, multiple genes that pretty much all do the same thing. So if one of them is defective, no big deal because you've got basically a backup. More so than the fact that you have 2 copies of each gene. In this case, you have 2 copies and 2 more copies and maybe even 2 more copies, right? So it's redundant. So this occurs, of course, when you're talking about more than one gene, right? 2 or more different genes influencing the outcome of a single trait. Essentially, really, pretty much all the traits that we look at are really a combination of many genes, right? So in truth, most of what we look at are gene interactions. Nothing is really truly on its own. So morphological traits such as height, weight, and even pigmentation are affected by many different genes in combination with environmental factors. So when we were looking at, say, the color of the pea plant, it isn't truly just a single gene that determines whether it's white or purple. Through further research, right, they found out that there were multiple genes involved. It just happened that there weren't any differences within the populations. They were so purebred that Mendel worked with that they only varied on one set of those genes. And so that's why he was able to study it in that way, where in fact there's I think 9 different genes, right, that actually affect the color. And so if that doesn't make sense to you, I think later on there's a little diagram to help us out with that. So good little summary table of each of the 4 that we'll talk about as it relates to gene interactions. So epistasis is inheritance pattern in which the alleles of one gene mask the phenotypic effects of the alleles of a different gene, right? So it overrides, you could even say, the other one. And this will make sense when we look at some specific examples. Complementation is a phenomenon in which 2 parents that express the same or similar recessive phenotypes produce offspring with a wild-type phenotype, right? So if they're both recessive, you would expect all the kids to be recessive, right? Like, okay, well, how are they? Basically, the dominant trait, the wild type that we see most often, right? How is that possible? Well, because we're actually talking about more than one gene, right? If it was really one gene and both parents were recessive, then the kids would have to be recessive, right? That makes sense to you guys? Again, we'll go over some examples. Gene modifiers, a phenomenon in which an allele of one gene modifies the phenotypic outcome of the alleles of a different gene, right? So again, this is really true interactions where one affects the other. And then redundancy, as I said, is in a pattern in which the loss of function in a single gene has no phenotypic effect, right? So the fact that you don't have this one gene is no big deal because you have other things that pretty much do the same thing for that phenotype. But loss of function of 2 genes has an effect, right? So, okay, you don't have either one of these genes that do this phenotype, so therefore you don't have that functionality or that phenotype. So that is on the premise that functionality of only one of the 2 genes is necessary for the normal phenotype, right? You don't need all of the copies that you have to all work. You need at least 1 to, right? And you will have the dominant phenotype. So when we're looking at 2 gene interactions, you can still produce 4 distinct phenotypes. Like we do when we looked at 2 traits, right? You can still, even though we're looking at 1 trait, we get 4 different expressions of that. So the example they give is a classic one of the comb morphology in chickens. So that little thing on the top of their head, it's called the comb. And so it was discovered in 1906. And you'll notice one of the guys, Reginald Punnett, right, is responsible for the Punnett squares. And then Bateson was his co-contributor. So there's 4 different morphologies for the shape of the comb. So you have the rose comb, and you have the pea comb, and then the heterozygote is a walnut comb. And then you have the single comb, which is actually the recessive of both rose and pea genes. So depending on what combination you have, of the 2 genes will determine your phenotype. So, all right. I want to remember something. So if we generate a heterozygote for the walnut combs, where they have a dominant and recessive allele for each gene, right? For rose and pea, they're going to be all walnuts, right? And then when you do a cross between those individuals that are all heterozygous for both genes, we end up with that big 16 square Punnett square. Do you guys remember what the ratio is that we typically see with 2 different traits when we were doing our Mendel crosses of a dihybrid cross? 9, 3, 3, 1. Do you see that here? Yeah, look, we end up with 9 walnuts, 3 rose, 3 pea, and 1 single. So the F2 generation, right, of the 2 heterozygotes, we end up with that ratio we've seen before for dihybrid crosses, right, of 9, 3, 3, 1. And that's again because we're looking at 2 genes. But in this case, just a single trait, right, the comb. Where before when we were looking at it, we were looking at 2 separate traits at the same time. Remember that? Right? So like round and yellow peas as opposed to green and wrinkled. Right? So you got those 4 different combinations of those 2. So we still end up with that same ratio, but we're just talking about a single trait now. But it's 2 genes, that's why we end up with this same ratio. I'm doing facial recognition right now. Okay. Some people won't even look at me right now.
So do you have genes where there's only 2 outcomes? I mean, 'cause there's 4
Yes,
outcomes
yes,
here
yes.
that matches that cross.
Yeah, you can. Yes, you can. With the color thing that we got coming up.
Okay.
Yeah. Okay. So, but this is why, for them, when they looked at this, they're like, it can't be just 1 gene, right? Because we got 4 different possibilities here. It's probably 2 genes, because look, we got that same ratio that you get for a dihybrid cross of 9, 3, 3, 1. You see the relationship there? Similarity? Okay. So, I think that's, all right. So, R for rose comb is dominant over little r, right, which would be recessive. P is for P comb, which is dominant over little p. If you have one R and one P dominant ones, it's said to be codominant, so both expressed, and that's why you end up with the comb that looks like a walnut. And then all recessives for both P and Rose has that single comb morphology or phenotype. So epistasis is when the alleles of one gene mask the phenotypic effects of the alleles of another. And so this is relative to a particular phenotype. So Mendel's laws of segregation and independent assortment are still holding, right? They're, the parents, it doesn't matter. They randomly give whatever chromosome they give to their kids. And these two things are not on probably the same chromosome, so they're separating independently. If they were on the same chromosome, we wouldn't get this 9331 pattern. They'd be linked to each other. So they may mask phenotypes, but the 9:3:3 ratio of genotypes is still present. And again, this is relative to a particular phenotype. If possible, the wild type phenotype is the reference, right? The most predominant within the population is the wild type. So a cross involving two gene interaction can produce two distinct phenotypes. See, I told you it was coming. Right? So you can still have two genes and yet still only have two phenotypes. So the flower color, come to find out, actually is, more than one, right? So say it was two. So Benson and Punnett worked with these plants further and attained several true breeding varieties of white flowers, and they carried out the following crosses. True breeding purple with true breeding white, and they got all purple flowers, just like Mendel did. And then the second generation, right, got back the white, and the typical ratio we expect 3:1, because purple is dominant. The results were not surprising, right? Until they did a cross of some of the purple heterozygotes and actually got half and half. So, well, not half and half, a 9:7 ratio. So this is what they expected, right? So they reasoned that it wasn't a single gene, right? That it maybe was determined by two different genes. And that's how, if you wrote it out like they did in that Punnett square, how you could end up with that ratio that they got. So they surmised that one gene that they're denoting C determined whether the plant could even produce color. And it was dominant over not producing color, which of course we know is white. P, another color producing gene, but in this case, the purple color, another purple color producing. They're both purple color. But one they denote C and one P. It'll make sense when I get to the next thing. The recessive of either is masked by either one of the dominants. Thus, a homozygous of either recessive allele would develop the white flower. regardless of whether the other gene contains purple-producing allele. So the problem with this, and what they believe could be the situation, is at the molecular level, you have some compound, right, some precursor that doesn't have color. And you have some enzyme that converts it into an intermediate that also still doesn't have color. And so then it takes the next enzyme to actually generate the purple pigment. So if you don't have either one of these, you can't get to the end, right? So if you're recessive, which means basically it's a loss of function, it's an enzyme that doesn't work, for either one of these steps, you're not going to make it to the end. Does that make sense to you guys? Right? So enzyme C is needed to go from here to here, and this other enzyme, say P, right, coded for that P gene, goes from the intermediate to the purple. You got to have both of these in order to actually be purple. Right? So if you have recessive alleles, you don't have this enzyme, you can't go from precursor to intermediate. There's no way you're going to be purple. You're going to be white. If you have this one, but then you don't have this one, again, you can't turn this intermediate into purple. So you're not going to be purple. You're going to be white. So either recessive, homozygous recessive for either enzyme results in no production of purple pigment. So when you have, let's see, is this our next one? Oh, it's the same one. So this is referred to as complementation. So you have two mutant lines, right? Two different enzymes that you could have a non-functioning enzyme that affect the same phenotype. You can have three distinct phenotypes due to epistasis. So inheritance of coat color, right? What their fur coat looks like for rodents. True breeding black rat crossed with a true breeding albino results in agouti. That's how you say it. Agouti? I don't know. Coat color. This is pretty. It's where at the tips they're black, but at the base of the hair it's orange, right? So this is where they go from orange to black. It's kind of cool. So if the agouti animals are crossed, they produce offspring with the following ratios: 9 agouti, 3 black, and 4 albino. And so the way the inheritance works for these guys is A stands for agouti, C for color, actually having color. And the agouti causes that shift from black to orange at the tips. So AA results in black throughout the entire hair. So the agouti, that having that ability, keeps it from being completely black, right? It's orange and black. The C determines whether it'll be pigmented at all, right? So if you don't have the ability to be pigmented, you're going to be albino. You're going to be white. So So if you're recessive for color, you're going to be albino. So when you do a cross of two heterozygote agouts, you end up with that ratio that they just said, the 9:3:4. So at least one copy of each dominant allele results in the agout color, right? So if you have at least one capital A and one capital C, you can be colored and you're going to be agout, right? If you don't have color, right, if you don't have the color dominant, you're going to be white, you're going to be albino. If you're recessive for agout, right, you don't have that ability to change to the orange at the root, you're going to be solid black. So it's a modifier, right? Agout. So in some cases, two genes may interact such that the alleles of one gene modify the phenotype produced by the second gene and instead of masking it. So that's the next thing we're going to look at. So inheritance of creamed eye allele in Drosophila, and this was discovered by Calvin Bridges. He identified a rare fly with cream-colored eyes in a true breeding culture of flies with eosin eyes. It's kind of a reddish color, but not like a true red. This can be explained in one of two ways. A new mutation, right, happened and changed the eosin into a cream allele, or a mutation occurred in another gene that modifies the expression of the eosin allele, right, and affects that other allele. So the hypothesis they came up with is that creamed eyes in fruit flies are due to the effect of an allele that is the same gene as the eosin, or in a second gene that modifies the expression of the eosin. So then they tested their hypotheses, and they crossed cream-colored males with wild-type females and got all red eyes. And then they took a brother and sister of that population, and they ended up with 104 females with red eyes, 47 males with red eyes, and 44 males with light eosin eyes, and 14 males with cream-colored eyes. Why are they mentioning male and female? Yeah, so it must, the sex chromosomes must be coming into play, right? If you look at their diagram here, notice X, X, Y, Y. One of the genes is on the sex chromosomes. So the sex also determined. So this would be, no, is it influenced or limited or I don't want to say wrong. I'll straighten it out in my brain in a minute. Okay, so it contains males with the light eosin eyes. This indicates that the cream allele is not in the same gene as the eosin, right? So this isn't a mutation. The fact that we got this new phenotype here. So one possibility that eosin is an autosomal recessive allele. Autosomal meaning, again, it's not in the sex chromosomes, where the other eye colors are on the sex chromosomes. It's X-linked. Eye color is X-linked. That's what it is. It's X-linked because it's on the X chromosome. Did you notice that it's not on the Y? Okay. Hopefully that will be coming up. Okay. Does not modify the eosin phenotype if the allele is normal, right? The recessive allele generates that cream allele, and because it modifies the eosin into what we refer to as the cream color. Has no effect on the red eye allele though, so it just affects the eosin. So when we look at this, right, so the big C's are denoting that modifier, right? And let's see, they have the code over here for us. Any fly with XW plus has red eyes. C or CA have no effect, right? So because remember, it doesn't modify red, right? And that's the wild type. That's why they put W plus, right? That's why it's written like this. So this is the wild type. These are all red eyes. Y'all see that? Or it's kind of pink here. I think they did pink instead of red because, right, red would be hard to see. Pink is just a lighter shade of red. Okay. Any fly with the XWE is eosin, right? So that's letting you know that it's the eosin. As long as there is one dominant C, it doesn't get modified, right? It's when it's in the recessive form, the CA, then you have the modifier, and you end up with the cream eyes. So there it is right there, right? Where you have the recessive for the C, and you have the eosin on that X chromosome. Where here, because I guess it's a male, right? And that's eosin, but you have one of the color ones. It ends up being that light eosin instead of the really true eosin. So what do you think, what type of inheritance pattern do you think we're maybe seeing here? So this is where they only have one X, right? So they're making that color with just one X. Where if they had two X's, like this female, let's find a female with eosin. Do we even have a female with eosin? Eyes. I don't understand how they have the chart. That's the light eosin, right? Wish they'd write out the ratio next to it. Where's our data? Let's go back. Oh, all the females are going to have red eyes. That's right. That's why. All females have red eyes. Because red is dominant. And since they have two X's, they'd have to have two E's, and none of the females have that. But the males can end up with a single E because they have a single X. And so if you think about that, why these guys are lighter is because they're not producing as much of that pigment. So what would that be? What type of inheritance pattern? Well, this is X-linked, but also it'd be kind of like incomplete, right? Because it doesn't produce enough pigment to give the true eosin color. So this is like the 4 o'clock plants, right? Where you have the pink flowers instead of the red flowers because they're heterozygous. And so the males are kind of like heterozygous because they only have one copy of the gene. With me? No? Kind of, sort of, maybe. We'll move on. All right. So females are all red because they all inherited the wild type red, which is dominant. The males, as we saw, can have red eyes, or if they have just the eosin, they can have the eosin, but they're lighter, right? Because they have less of the pigment being produced. Is someone knocking on our door? Locked again. All right, and then you get the cream, right, when you have that recessive color modifier. modifying that eosin to the cream color. It can, and again, as we said, it affects the eosin, but it doesn't affect the red color. And only the homozygote. So they, of course, the predicted ratio agrees reasonably with the data that they got. If you were to do a chi-squared, right, on their data, it would match up. Okay, so designers develop techniques to detect directly generated loss of function alleles. These are known as gene knockouts, and this is used a lot in research. And so it really helps us know how a gene functions when we take away that ability for a cell or an organism even. So that's what we do. We literally, we mutate that gene, basically knock it out, make it non-functional. Does that make sense to you guys? So there is actually a line of mice that one of the researchers, when I was at LSU doing research at LSU Vet School, that one of the immunologists worked with, and this was a really expensive line of mice. When they would talk about the knockout mice all the time, I was, they were concerned about, the caretakers taking care of them properly and everything like that. And I was like, what's the big deal? I really didn't grasp because nobody explained to me what knockout. I mean, I had an idea. I was like, knocked out means they don't have something, right? Most people could surmise, okay, well, they don't have. Yeah, they had been genetically specifically modified to not have a particular gene ability, which is not an easy task. And especially that was, okay, aging myself now, like 16 years ago that I worked there, right? So that was a big deal, right, to have that particular line of mice that had that particular mutation, because she was studying, a particular immunological, I don't even remember what her research was on, but it was pretty phenomenal. She was pretty amazing. And now I do understand why they were so concerned about these mice, right? Because it's impressive and important thing to be able to do. So the thing is, too, is that a lot of times, too, you don't really see it in the phenotype, right? We're talking about at the genetic level, right? Not, and even in the molecular level. So I am gonna try and look up her research because I think it'd be cool to have like an example and something that I never got to learn about when I was there because I was busy doing other things. So I'm gonna rewind the clock, right, and give you guys a real world example. I'm gonna try and find her research. So I think it'd be cool to talk about. And I'm sure if I contacted her too, she'd be willing to maybe even come in and talk to us. She's still at it. So we were working on problems. And I've posted some for you, like I said, on Connect that you can do multiple times, right? Unlimited, get all your points. And we'll go over them next week after we lecture on Chapter 6 stuff. And I should have that PowerPoint up for you guys by definitely the end of the weekend. What do you mean 12? Oh, slide number 12? Problem number 12. Yes. Thank you. So this is a good one too. This is a good one. So in shorthorn cattle, so this is concept question number 12 from the back of your chapter. So let you know what the C12 stands for if you forgot where I got these from. And I think this one they actually give you the answer. Let me pull up the book. Because it's the evens, right, that you get the answers to. And so chapter 7 and concept 12, yeah, so it gives you the answer, but That doesn't always mean you understand it, right? So I felt this would be a good one to go over. Okay, so in these cattle, coat color is controlled by a single gene that can exist as the red allele, right? So R, capital R, so they're telling you it's what? Dominant. Or white allele, little r, so that's recessive. The heterozygotes, though, right, have a coat color roan that looks less red than the RR homozygous. Okay, so what does that sound like to you guys? What pattern of dominance right away? Is it incomplete, co, or something else? You think it's codominant? Anyone else? So when you're saying codominant, what do you think's happening? Trying to fight for dominance. But codominance means what though? It's not fighting. What's our prime example that we always remember for codominance? What'd you say? The red blood cells, blood typing, right? So what's the codominant blood type? AB. So what's happening there? Both are expressed, right? So you're saying both red and white are expressed. If you're saying it's codominant. Oh, you're reading ahead. Are you reading ahead? Oh, okay. No, it's okay if you are. Okay, so it says, however, when examined carefully, the roan phenotype in cattle is actually due to a mixture of completely red hairs and completely white hairs. Right? So some of the hairs are completely white, others are completely red. So when you look at the coat, right, you just look at the cow, it has this color that's not quite red, but obviously not white. It's what they call roan. You've probably seen it. If you've seen a cow, you've seen it. Some people say it's orange or something or whatever. Or, I don't know. So if you're just talking about the coat itself, at that level, you would say it's codominant, right? But if you are actually looking at the hair level, what's going on, right? Some of them are red and some of them are white. Shouldn't they all be what? Should all be, for dealing with standard dominant recessive, they should all be red. So the reason they give, I didn't copy it. Why didn't I copy it? Maybe it wouldn't let me copy it. Okay. And this is like insanely small. Yeah, it won't let me copy it. I should have went to my instructor's guide. Okay. It's insanely small. I apologize. Here, I can blow it up, can't I? Hopefully my pop-up will blow up too. Okay. Just gonna move over more. All right. So perhaps it should be called codominant at the hair level because one or the other allele is dominant with regard to a single hair. However, it's not the same as codominance with blood types, right? In which each cell can express both.
The hair cell only expressing one and the blood cells
Or but be able to express both.
So it's not really codominant.
So it's not, I mean.
So it's really something else.
Yeah, it probably is something else. And this is a good, I'm like, really? But this is one of those things, remember where we said, what's happening at why it says sometimes what's happening at the molecular level, right? So at the level of, what determines the color of the hair, right? Like we said, like we were dealing with, normal patterns of dominant recessive, we would expect them to all be red, right? But for some reason, some of them are being red and some of them is being white. So we're going to get to a phenomenon in this Next chapter coming up. I hope it is the next chapter. It should be. Which I think might have this example make a little bit more sense. And I wish they had more of an explanation there, and shame on them for not. And they might just not know it. I mean, we might not know down to the molecular level what's going on with this coat color thing. There is a possibility we don't know. So I wanted to skip ahead to what I think is the coolest. Is this? Okay. I need to not be so big because it's messing me up. I want to go to chapter 6. Is that right? No. We're not doing 6. We're doing, what would be next? 5. I don't know why it's 6 on the brain. 6 on the brain. So, are we not going to do it? That's imprinting. Oh, maybe it's in a later one. I don't want to spoil it for you guys yet. Or maybe I do. Calico cats. Anyone know about why calico cats are calico? Anyone know what a calico cat looks like, first of all? No, it's actually a calico cat. If you see it, it's a female. Can only be a female. Let me put it this way. If it's a male, it's sterile because it has 2 X chromosomes and a Y. It's having the 2 X's that you can have the different patches of orange and black. Orange and black are on the X chromosomes, and each cell has to pick which X chromosome it's going to use, and so that's why some of them are black and some of them are orange. Cool, huh? So for women, we shut down one of our X's for the most part. Becomes what's called the bar body. So it's determined by which X you use. So in that case for calico cats, that's why they can be orange or black, because there's either orange on an X or a black. So it might be something like that, like, how is it being expressed? Like, it might be that it's on that X chromosome like it is for calico cats, so that might be why they're shutting down the one that has it, and therefore the other one that doesn't have it, they're not making it. Does that make sense to you guys? Going too far ahead, maybe. Okay. I think it might be something like that. Okay. So questions. So this is the one I stole from Canvas. That's why it doesn't have a title. I mean, Connect. I took it from Connect, chapter 4. Connect, chapter 4. Okay. So, a man and a woman have 5 children. What is the probability of this couple of having 4 unaffected, right? So this is where they have the good hemoglobin versus the sickle cell hemoglobin, where if they're heterozygote, remember, they have at least 1 copy, and that's enough to not have sickle cell. And 1 affected child if both parents are carriers. So let's back up one more step. What type of dominance pattern are we looking at here with sickle cell? It's not codominance, not incomplete. It's one more dominance, where it's beneficial to be the heterozygote. Overdominance. So this is our overdominance example. Okay. So, how would we solve this? Product rule or binomial expansion? Set of 5. Yes, 5. It's a set. Yes, you guys got it. It's a set. Binomial expansion. Irrespective of anything. right? That's why I grabbed this one, because again, I didn't want to make you guys calculate a binomial expansion to be able to do it and connect. You guys recognized it. See, you're getting it. You're getting it. It's taking us a while, but we're getting it. So yeah, there's my Higgins law. Binomial expansion. Okay. So I didn't steal any more yet. Oops, I didn't go to the end.
So on the test, is it going to be like, as an example, then is this like codominance or X-linked? I mean, is that
Yeah.
determined based on examples?
Mm-hmm. Yep. Probably do it like a matching. Match which one's over dominance versus co versus incomplete. Because one of the objectives is to be able to compare and contrast, right, between those. Okay, so we did 12, 13, what's 13? Ooh, vitamin D. X-linked. Dominant. Want to work through 15? Let's see. Copy. New slide. C15. I think this might be the only one we'll have time for. Okay. Let me make it big so we can all read it. So, a human disease known as vitamin D. And the thing is, y'all, I'd have to give you enough information that you could figure it out, right? For examples, right? You should know the main examples we use, like the codominance blood types and the 4 o'clocks for incomplete and sickle cell for overdominance, right? A human disease known as vitamin D resistant rickets is inherited as an X-linked dominant trait. Again, I'd have to tell you that, right? Since we didn't go over this particular disease, right? I'd have to give you that information. So remember how before the problem I had with one of those problems is they didn't tell you guys that Huntington was, I think, rare meant that usually most people are heterozygotes for that dominant trait, right? So I won't do that to you. I will give you the information you need. Okay. So if a male with the disease produces children with a female who does not have the disease, what is the expected ratio of affected versus unaffected offsprings? So, okay, it's dominant, right? It's X-linked. So a female who does not have the disease, right? So it's dominant, so that means that both her X's are recessive. And a male with the disease, but he only has one X, right? Did we do sex linked yet? Do I have any X's and Y's I can steal? All right. So first we got to do our X, and we're going to put, what is it, rickets? Let's do a capital R. All right, I'm not going to bother with the subscript right now, y'all. Don't drive me crazy. I'm just going to put it next to it. Superscript. Up, up. All right, so little r's for her, right?
Does she get a big X and a little R?
No, X's don't get size differences, right? They're just sex chromosomes. It looks what? Oh, like a big X? Yeah, it's just because it auto does that. Yeah. Okay. So, she would be big X's too. Okay, so big X's, right? And then heterozygous. Big X's. So females, as some of you guys I think I had already heard you say, are going to be, so that X only has one R. Should we put them next to each other? I guess I should do that, huh? R, X, R with big X. All right. So because, did I do that right? Yeah. Okay. Make it bigger for you? Okay. So here's the male, right? He has it, so big R, right? And nothing on his Y because it's X-linked. Here's mom. She doesn't have it, little r's, right? So she gives all little r's to all her kids, right? And her son is lucky. Her sons are lucky because dad gives them Y's that makes them boys, and mom doesn't have the defective on either one of hers, so all her boys don't have it, right? But the bad news for the girls, because they're a girl, right? They got their X from who that makes them a girl? Their dad, which has it, and so all the girls have it. Make sense? Bad news, but
What was that?
yeah. And usually they give us real examples, like it's not fake. Vitamin D deficiency. It's a vitamin D deficiency. The good news is that one's really easy to fix, y'all. Vitamin D supplements. I even take those.
I thought it was vitamin D resistant rickets. Then you wouldn't, right?
Well, I don't know.
Is
I don't know. It says resistant, so what does that mean?
So then you can't take vitamin D because it won't do anything, right? Or like if you take vitamin D, it's not gonna. Yeah. Right? It's resistant to like isn't vitamin D what makes you not have rickets?
Right.
So you would get rickets because no matter how much vitamin D you have.
Oh, yeah, you can't process it. You can't process it to what you need it for. Yeah, I don't know. We'd have to look it up. It's another one of those diseases someone could pick. Email me and it's yours. All right, I'm going to stop there. I don't think anyone will complain, huh? We only have 3 minutes left anyways.